Welcome to my presentation, which is titled The Interplay Between Diabetes and Cardiac Metabolism. This work was done in collaboration with the University of Leeds and is part of the final year project biomedical engineering course at King's College London.
This is the table of content, and let's get started. So, what is diabetes? Diabetes is a metabolic or autoimmune disorder characterized by high blood glucose. This is caused by insulin malfunction, resulting in numerous metabolic disturbances.
These disturbances affect the body's energy utilization, altering its functioning.
Diabetes mellitus is one of the most common forms of chronic diseases worldwide. It is estimated to have affected 422 million people globally as of 2014 and caused around 1.5 million deaths in 2012. The estimated cost for diagnosed diabetes in the US has reached $327 billion in 2017. High blood sugar levels can damage the large blood vessels in the body, including those in the brain, legs, and the heart. This can lead to cardiovascular disease, a common issue in diabetic patients and a contributing factor to roughly 70% of their deaths. Ischemic heart disease and congestive heart failure are among the most common CVDs linked with diabetes. Cardiovascular disease development is complex and intricate and is influenced by several factors including abnormal glucose and lipid levels and/or blood pressure changes. These factors foster structural and pathological alterations in the heart, impairing its muscle tissue or electrical functionality, a condition termed as diabetic cardiomyopathy. This heart condition can affect the heart's size, shape, and thickness, thus impacting its function and performance. Diabetic cardiomyopathy leads to noticeable alterations in heart structure. Typically, research emphasizes two key structural indicators: well-recognized left ventricular hypertrophy, or an increase in left ventricular thickness, and concentric heart remodeling, or a reduction of chamber size. However, it is hypothesized that there are specific remodeling patterns associated with diabetes mellitus that expand beyond the well-recognized wall thickening. Therefore, in our research, we constructed statistical shape models from MRI data to better understand the remodeling patterns that differentiate our control from our diabetic hearts. This study's goal is therefore to provide new insights for improving cardiovascular disease management strategies in individuals with diabetes. Creating a statistical shape models from cardiac magnetic resonances images involves three main steps: semantic segmentation, 3D model fitting, and dimensionality reduction techniques. Let's go step by step at how each one of these are done. First thing we need is obviously the data. So in this study, we utilized and we analyzed two group of CMR images from two cohorts, one from the UK Biobank and another one from Leeds. Now, in terms of the design study, we performed one-to-one matching to compare left ventricular shapes across different cohorts between healthy and diabetic patients. The UK Biobank cohort statistical shape model included 132 controls and 132 diabetic cases matched based on age, gender, and BMI. The Leeds cohort is a statistical shape model that was developed using 13 controls matching with 39 diabetic cases, matching solely on age and gender. Lastly, the mixed cohort statistical shape model matched 45 controls from the UK Biobank with 35 diabetic cases from the Leeds cohort, using our initial matching criteria. This helps reduce bias and the effect of confounding variables when revealing the differences between the controls and diabetic groups across our cohorts. From our Leeds CMR data, our cardiologists then manually delineated the endocardial and epicardial contours. And for the UK Biobank cohort, we trained a NN unit segmentation network to segment automatically the left ventricle. Now, having the raw data, how do we create our individual meshes? We start with a predefined high-quality cubic Hermite mesh template, which essentially is a one-size-fits-all heart. We perform image registration and variational warping, which allows us to go from the predefined mesh to a smooth patient-specific left ventricular mesh. We use the statistical tools PCA and LDA to make sense of all the 3D models of hearts we created. So, what are PCA and LDA? Briefly explained, PCA is a technique that helps us reduce the complexity of our data. From our many heart models, it identifies the key patterns or modes of variation, which best explains the direction of variation. LDA, on the other hand, is a technique that takes our PCA results and tries to maximize the differences between the heart models of diabetic and non-diabetics. It's sort of like a way to super filter and emphasize the heart features that really matter when distinguishing a diabetic heart from a non-diabetic one. Bear in mind that what we see within the center of these shapes, colored in orange and in blue for the PCA and in purple for the LEA, is the mean left ventricle shape. We use this in order to help us infer the patterns in the 3D models. Now, let's look at our results. Let's look at the heart features that best distinguish a diabetic heart from a non-diabetic one. These plots are created from our LDA analysis, and this is a view called the thickness map, which visualizes heart thickness via color, from red, which is more thick, to blue, which is less thick. Additionally, we include the HAA 17 segment plots, which provide a flat view of the different heart segments. The white sphere shows the middle of the septum, and the black sphere points to the anterior of the left ventricle. As we can see, the left ventricular walls of diabetic patients, towards -3 standard deviations, are generally thicker. However, the apical walls show no thickening, and the control subjects have thinner left ventricles. If we look closely into the diabetic heart with respect to the mean shape changes, its eccentricity is exhibited through the bulging of the posterior left ventricular wall. These eccentric changes mean that if we slice through the heart, the size of the heart's chambers is enlarged or dilated in the mid walls with respect to the basal walls. For the Leeds cohort, our diabetic patients, +3 standard deviations, exhibited thicker left ventricular walls. Again, the apical walls show no thickening. Non-diabetic subjects show thinner left ventricular walls, which is completely expected. If we look closely into the pattern of the diabetic cases, there's an inward remodeling towards the anterior of the left ventricle, as seen due to the black sphere. This contradicts what we have seen previously, as this eccentricity change is completely opposite to what we've seen for the UK Biobank cohort. These conflicting patterns observed in the UK Biobank and Leeds shape statistical models led to us create a mixed statistical shape model using UK Biobank controls and Leeds diabetics. This was solely done to determine if the differences that we have been seeing is due to variations in the controls or the diabetic groups. However, we still found out that the diabetic patients show general left ventricular wall thickening, whereas the control subjects show substantial thinning of the posterior left ventricular walls, which just contrast that our results are consistent. Now, what can we draw? What conclusions can we draw from that? Now, we can draw that there's a shared left ventricular thickening deformation across studies, but differences in PCA mode variations. Left ventricular wall thickening and reduced left ventricular volume is seen, and concentric remodeling are very common with diabetes. These changes in eccentricity of the left ventricle have also been observed, and this eccentricity changes explain that the heart works harder, but because there's distinct patterns, we can hypothesize that the diabetic heart is able to adapt in two distinct ways, which means that there's two diabetic phenotypes, with the most extreme of this belonging to the Leeds diabetics. Now, we've had several limitations through this study, with limitations due to using linear machine learning methods such as PCA and LDA, which present limitations in analyzing complex structures like the human heart, which may not always lie in a linear subspace. Also, we've gone through several lack of diversity in the datasets, being a significant limitation in the study, since there's underrepresentation of various ethnic groups, with mainly the white background being represented. Another issue is the orientation scissors between different imaging formats, which flips the images 90 degrees one to another. Now, developing our statistical shape models, we have achieved sub-voxel accuracy in fitting the meshes, and our findings indicate that 3D left ventricular remodeling in diabetes mellitus involves not only well-recognized left ventricular hypertrophy and concentric heart remodeling, but it also is accompanied by eccentricity alterations. Further research needs to be done in order to better identify and understand these eccentric adaptations. But one thing that can be said is that the understanding of these adaptations opens up potential avenues for re-evaluating clinical approaches. For instance, allowing clinicians to know how a diabetic heart is adapting to the disease as it is progressing. Thank you very much.